All right, so our B cells have undergone clonal selection, clonal expansion, and we have produced plasma B cells that are churning out these antibodies, 200 of them per second. Okay, so what do these antibodies actually do? Why would we be producing so many of these so quickly? Well, they're actually quite a cool protein, to be honest. Some of the main things that they're doing, we've actually seen a couple of these things happen before, but remember, these antibodies have a specific site on them. So they're specific to a unique non-self antigen that a particular pathogen has. So it will only target the pathogen that has that corresponding non-self antigen. It won't target anything else. So a couple of the things that our antibodies can do once they have been produced by the plasma B cells, they will be circulated through the entirety of your body in the hopes that they will randomly collide with those non-self antigens. So the first thing that they can do is something called agglutination. So agglutination occurs when you have a clump of antibodies that essentially form a clump or a network between those non-self antigens or those pathogens as well. So here is a diagram of agglutination that is occurring to a toxin. So this might have been a toxin that a bacterial cell has released. And because multiple antibodies are clumping multiple non-self antigens together, this is a form of agglutination. If you watched the video that was on the previous slide, you would have also seen an example of agglutination happening where multiple antibodies were holding multiple bacterial cells together in a clump. That's an example of agglutination on a cellular level where they're holding multiple bacterial cells together and preventing them from further spreading throughout your body. The other thing that our antibodies can do is opsonization. Okay, now we've heard this word before. This was a word I introduced you to when we spoke about complement proteins. So the same meaning that it had in complement proteins, it now has for antibodies as well. So opsonization of a pathogen will occur when an antibody, or several antibodies I should say, completely covers the surface of that pathogen. So we've got it shown down here in this diagram, but we've also got it shown here in this diagram, where the antibodies are completely covering the surface of this pathogen. And that'll do two things. The first thing it will do is it's going to flag our phagocytic cells to come along and engulf this pathogen. But it will also mean that it's created this protective layer that's insulating the pathogen, and the pathogen can no longer do damage to the body. So it's kind of got two benefits there. And the last thing that you should be aware that our antibodies can trigger or cause to happen is they can cause our complement proteins to form that MAC complex, which was the giant hole that punches the side of a pathogen's plasma membrane and causes fluid to rush in and eventually causes that cell to burst. So that's, I guess, another feature that your antibodies can trigger in your body. One thing that I want to make really, really clear is that these antibodies that have been produced by your plasma B cells, antibodies do not directly kill a pathogen. So they do not directly kill a pathogen. They flag, they trap, they signal other things, and those other things will come along and then destroy the pathogen. But antibodies do not destroy pathogens. They cannot kill a pathogen. Please just keep that in mind, because a lot of students will automatically assume that just because cytotoxic T cells have the ability to destroy cells, that B cells do as well. B cells do not have the ability to kill pathogens. They produce antibodies that assist and neutralize them, but it is other cells of the immune system that will ultimately lead to the destruction of that pathogen. Now that we've heard what an antibody can do, you are required to know about the basic structure of an antibody. So I've spoken to you about it being that basic Y shape. It's a little bit more detailed than just a basic Y, and it is important that you do recognize the features of an antibody. So we're going to start off with what makes up the Y. Okay, so there are, I guess, three main structures that you need to be aware of. If we're looking at the diagram that's been provided on the screen here, these purple sections are called heavy chains, okay, and they make up the bulk of the antibody. Probably about, I don't know, 2/3 of it. The section in yellow, okay, so there's two sections in yellow, one here and one here. They're called the light chains, and they make up close to about 1/3 of the antibody. Okay, so we've covered the purple bit and the yellow bit. The purple and the yellow bit, so the heavy chains and the light chains together make up what's known as the constant region of the antibody. Okay, so constant region, which means that it's pretty much going to be the same no matter what cell you look at. It's constant. The part of the antibody that's going to change between cells or between B cells that are producing them is known as the antigen binding site. And the antigen binding site is the section in black. So you can see that there's a section in black on this arm of the antibody, and there's also a section in black here. Okay. So these antigen binding sites are the parts of the antibody that will change depending on what cell has produced this antibody. It is the part of the antibody that is complementary to the shape of the antigen that it is targeting. Okay, so you can see in the example on the screen here, the antigen that we're targeting is square in shape, okay, which means that we obviously need to have an antigen binding site that is complementary to that shape. And in this diagram, we can see that that's the case if I just rub that stuff out there. Please take note that any antibody, both of the antigen binding sites, so both of the sections in black on each arm, will be identical for that antibody. Okay, we're not going to get something that's complementary to the square-shaped antigen over here and then something that's complementary to the circular one over here. No, never going to happen. Both of these sections need to be the same. If you want to see some other, I guess, visual representations of some antibodies, there's a couple on the bottom of the screen here. Okay, so you can see that, again, the basic structure of the antibody, so what would be the heavy chain and the light chain making up the majority of the Y, is consistent across all of those antibodies, antibody A, B, C, and D. But as you get to the tip of the Y, at the two arms, you can see that they are different shapes. So we've got different antibody A. They are consistent across antibody A, but they are different to what would be on antibody B. And again, different to what you'd see on antibody C. So those regions that I'm colouring in now, they are representing the unique antigen binding sites that would be interacting with a specific non-self antigen that's found on a pathogen. So the non-self antigen or the pathogen that antibody A would target is completely different to what antibody B would target, or what antibody C would target, or what antibody D would target. Okay, and each one of these antibodies is produced by a completely different B cell. Okay, they will not be produced unless that particular B cell that has those specific antibodies on its surface interacts with the non-self antigen that is complementary to its antigen binding sites, where it undergoes clonal selection, then clonal expansion, and the plasma B cells that are produced from clonal expansion would then start to produce that specific antibody en masse. Okay, that's the only way these individual antibodies are going to be produced. So just to quickly recap, we have our Y-shaped structure made up of 2 heavy chains, which were the ones in purple, 2 light chains, which were the one in yellow, and each arm of your Y has the unique antigen binding site, which is in black. Okay, and that will stay consistent across both arms of the Y. It will only change between antibodies that come from different plasma B cells. And finally, just reminding you about our memory B cells. So these are also produced once the B cell undergoes clonal expansion. They don't play an active role in the initial stage of infection. They are there to be stored for future infections. Similar to how we produce memory cells for our helper T cell and our cytotoxic T cell, those memory cells are there so that if this same pathogen ever enters again, we will have a quicker and stronger response because there are more B cells or T cells with those corresponding receptors or antigens ready to interact with the non-self antigen of the pathogen. I'd also like to just point out to you this graph on the slide here, which is representing another way of looking at how infections work in humans. So what this graph is trying to show you is the level of antibodies that are produced by you when you encounter a pathogen. So for example, you get infected by a particular bacteria here. It's past your first line of defense. Your second line of defense is trying to do its best to combat that infection, but unfortunately it's not powerful enough. Okay, by this stage here, this is where your antigen presenting cells have obviously started to digest some of the pathogen and are presenting that non-self antigen from the pathogen to your helper T cells, your cytotoxic T cells, and your B cells, ready to activate them or select them and then allow them to undergo clonal expansion. As that's happening and your B cells start to replicate, and then those plasma B cells produce antibodies, we're seeing the level of antibodies in our body increase until it gets to the peak, where that peak is reached when it's, producing the maximum amount of antibodies that it can, and that is usually enough to combat the infection and significantly reduce the numbers of that pathogen in your body. Obviously, when that happens, the tides have turned. Your body is now winning the war against this pathogen, and your B cells will slowly decrease the amount of antibodies that they produce. And most of those active plasma B cells will actually undergo apoptosis and be destroyed. However, we do know that your B cells also produce memory cells when they undergo clonal expansion. So your memory B cells, even though the free-floating antibody concentration has decreased, those memory B cells still exist and still have the ability to produce antibodies if activated again. And that's what's happening here, our second infection. So unfortunately, you're one of those people that, you get infected by the same pathogen again. Well, guess what? Your body has remembered it because it created memory cells, memory B cells, memory T helper cells, memory cytotoxic T cells. So it is not going to take as long from the site of infection to producing the peak of your antibodies. It's a much shorter amount of time. And you'll also notice that the peak is much, much higher. And that peak Well, that concentration of antibodies that is being produced by your body is so much higher because instead of just activating one B cell, which is what happened down here when you were first infected, your body had never encountered this pathogen before. This time, it's not just one B cell that is activated, it is all the memory B cells or all the memory T cells that are activated that are complementary to that pathogen. So you get like a tenfold, even more of a response that is faster and it's stronger, it's bigger. So usually on that second exposure to a pathogen, or even a third or a fourth, if you're that unlucky, your body is really quick at dealing with it, and sometimes you don't even notice that you're that sick. It's actually quite mild symptoms compared to when you were initially exposed, because it does take your body that much longer at the beginning to mount its immune response. We will also have a look at these graphs again when we look at vaccines and how vaccines work, but it's something to keep in mind in terms of the purpose of memory cells. They're so important because without them, we really would be struggling every time any pathogen infected us. Memory cells mean that we never forget. Our bodies know how to combat so many different types of infections, and they can do it so quickly, so fast, and in such a large number that most of the time we don't even know that we're sick when this occurs. It's pretty cool.